I think that right now, if you are not in China and heavily competing there and also collaborating there, utilizing China as a platform, you are probably out of the industry in biotech. So even like ChatGPT started doing the same thing. So somebody announces a huge round, and if you say, okay, who is number one? They're like, oh, this one, because it raised more. Like, why? If it raised more, it doesn't mean that it's better. That is why for us, we are not benchmarking ourselves against other AI drug discovery companies. It's nowadays irrelevant.
What is it? Fleming of St Mary's Hospital, London, who discovered that it produces the drug known as penicillin.
The question was, which drug would go with which cell?
It's a collaboration that's unexpected because it's between humans and the tiniest organisms.
The howl of a dire wolf.
Dorset lamb may represent a major landmark.
I did not start off planning to design proteins.
Scientific breakthrough will probably be something that we hard to imagine.
Test at birth could map out an individual's entire future risk of disease.
The first time I came across you, someone was talking about your tweets and that they were controversial, but that I didn't know why. And I still don't know why they're controversial.
Okay, so which one was the most controversial?
Honestly, I don't think your tweets are controversial. Like
some were, and I was kind of criticized for that, but nowadays I'm trying to balance, yeah.
What would you say are your most controversial opinions on Twitter?
Well, in some cases, I supported Elon's opinions, and Elon's opinions are quite controversial. But I actually think that, pretty much everything Elon does is probably for the good of humanity. So I like reposting his tweets in the context of our industry.
Yeah, that's not controversial to me.
But some people do find it controversial.
I know what you mean. I was definitely an Elon fanboy most of my life. I was worried that maybe he was becoming too political, but he seems to be a little bit more relaxed now again as well. I think he was best when he was like pushing his companies forward and competing with China. And I'm worried. I would be worried if he like had remained in politics, if those companies would have been able to compete, for example, against BYD or against sort of Chinese space companies, etc.
Good questions. I think that all of his companies are competitive, and I think that one of the reasons why he is successful is because he not only competes with China, he utilizes China as a platform. So if you look at his humanoids, I don't know what if you look at the bill of materials, how much of it is coming from China. My guess would be like 60% plus, even though he makes many components locally, but fingers, magnets, all the lower-end electronics. I would say without going into China, competing there, and also collaborating and utilizing China as a platform, you would not be able to succeed in a high-tech business today.
Yeah, I think this is one of the things that we're aligned on. And one of the parts about you that is like inspiring to me is the fact that the company is so global. It's headquartered in Cambridge, but you have sites in, I believe, in Shenzhen, or is it?
We have Shanghai, Yixing, Taipei, Hong Kong, Suzhou.
Okay.
So we are truly kind of Greater China. And we do have very significant presence. We actually kind of think of ourselves as a headquarterless company because many sites have different functions, and those sites may serve as a headquarter. So, for example, we have a site in the Middle East, Abu Dhabi. I actually think that will be the kind of center of the global renaissance for many industries, not only oil and gas and energy, but many industries, including potentially biotechnology. And whenever we're aligned with the project we're running, we can say that Abu Dhabi is our headquarters. I might also do something big in Saudi. Even though we are, of course, much of the senior executive staff in BD, in IR, in clinical development is here in Cambridge. The execution is on AI, the execution is in Montreal and Abu Dhabi. And then on drug discovery, we focus very heavily on China. As a matter of fact, just like in the Elon world, I think that right now, if you are not in China and heavily competing there and also collaborating there, utilizing China as a platform, you are probably out of the industry in biotech, unless you have discovered something that is a completely new therapeutic modality, like something that is truly innovative and where you can put your stake in and say, I invented this. And also in the area where it is difficult to fast follow, and also in the area where it's easy to automate, and also in the area where you don't need to have too many people working in the office. Because here in Boston, and if you look at kind of US-wide, people stopped coming to office as actively as they used to, maybe after COVID, or it's just a part of the culture now. In China, you cannot chase them out of the lab, of the office, and you actually have to introduce some policies so people don't work too hard, because in the innovative company, you need them to relax, and in some areas, you want to keep them very happy and innovating. But there, it's not about work-life balance at all. It's more about, when, at any cost, and when it's win against diseases, it's helpful for everybody. We're not in the industry that should be comparable to, let's say, TikTok or things that you can live without. In drug discovery, you probably should compare yourself to military. And it's not about work-life balance. It's about delivering the best therapeutic you can get on the market as quickly as possible, as cheaply as possible, as innovative of a therapeutic modality, and either small molecule or a biologic or whatever you discover, because currently over 60 million people are dying of aging and age-related diseases globally. That's more than in any war. So if you are thinking about, whether I should go and take a few days off that I didn't expect to have from the company, maybe you should think about people who are dying. And they're not dying in a fun way, right? So death is never fun. So regardless of whether it's a bullet or whether it's, pneumonia, in late age, or if it's ALS or Alzheimer's or cancer, it's terrible. And most people take it as natural. I don't think it's natural. It should be stopped. Many years from now, we're going to look back and think of us as a barbaric society because we didn't prioritize it. So I think that regardless of where you are, you need to prioritize biotech over anything. And if you are prioritizing, which bathroom to go to as a discussion topic instead of, how much progress did we make in biotech, that's a problem. That means that you are, typical hamburger diet and, TV warrior, couch warrior, talking about things that are may be important to some, but it's not important to everyone. And the topic that is important to everyone, it's aging, because all of us are aging, all of us are dying. It's going to kill you 100%. So there is no chance you can escape aging right now. There is no chance you can escape an age-related disease. So I think that the most important thing in the world is aging research. And right now we didn't even make a scratch. Actually, I did a fun exercise recently, and my hobby is analyzing government grants. So some people like to, go and watch Netflix. I can watch Netflix, but I also analyze government grants for many years. So my first exercise in this area were in 2007 to 2011, and I published a few papers on that, including the International Aging Research Portfolio, trying to trace the origins of biomedical innovation from the time you get the money to the time you get the product on the market. So, for example, in PD-1s, PD-L1s, or in certain gene therapy medicines, or in GLP-1s, how do you trace it back to the time the scientist got the idea, got the money, published the paper, got the patent, and took it all the way to the clinic and then got it approved and maybe even repurposed later? So usually it's not one person, but how do you kind of pass the torch? And we found some very interesting patterns. In 2011, we published a few of those. And then recently I asked LLMs, so pretty much every LLM, Grok, ChatGPT, Gemini, to summarize their internal understanding of what happened to funding and scientific productivity over the past 35 years, because now with LLMs, they know how to summarize, and that's where you can actually trace the origins better than just learning the patterns using, k nearest neighbors or SVMs the way we used to. So the question I asked was, what was the amount of funding that governments and the industry spent over the past 35 years? So starting 1990, 1995 to today, that is traceable, and how much life expectancy did we get in the US and globally, and also how many quality-adjusted life years on average did we get in the US and globally. And every one of those three kind of agreed that the amount of money that we spent was about $5 to $7 trillion, which is actually more than I expected and that I trace through my systems. And second, we got 1.1 to 2 QALYs over 30 years, 35 years. And in the US, we got like 1.7 years to life expectancy. And the funding has dramatically increased over the past 15 years, which is true. It's basically almost doubled. But the scientific productivity and the life expectancy gains and quality of adjusted life year gains actually have not dramatically increased. So nowadays, everybody is campaigning, oh, there are some funding cuts, and we're gonna die. No, you're not. You actually might get more efficient due to those funding cuts, because actually if you see the amount of funding that is going into projects that definitely will not result in a drug or some biomedical breakthrough that will help you live longer or improve your quality adjusted life years, it's massive. It's basically like, according to those LLMs, it's more than 25%. And it's increasing. So all the way from like 12% in the time when I started tracking.
Okay. And we're very ideologically aligned on the aging problem. I want to get more into, obviously, the beginning of Insilico and what is the cause of this decoupling, right? So historically, drug development costs have just been increasing substantially. Every single year, it's more expensive to sort of take a drug through clinical trials, and the success rate hasn't really changed despite the advancement in our tech. So what is the cause of the decoupling between funding and sort of success in drug development, and like why did you start Insilico?
Sure. So I started Insilico to go after aging, like in the most hardcore way possible. I am in aging research now for 22 years. So I had a pretty successful career in GPU, so in actually computer graphics. And I was very, very excited about GPU back then, but nobody expected it to work in AI at this level. So I worked for a company called ATI Technologies in the past. And all my life I was interested in aging. I just thought that I was born in Latvia. I'm Latvian and Canadian by citizenships. But when I was born, it was part of USSR. So when it collapsed, there was no room for science, so to speak. It wasn't even sexy, so to speak. When when the society collapses, some other values take priority. And there, people were very focused on making money and getting a name for themselves. But at the same time, computer science was advancing, very fast. So we lived through a period which is very similar to LLMs today, but in the internet era. And I saw so many things change. The bubble was very similar at that time to what it is today. And I decided to go onto the internet to make a little bit more money and become more independent from grants and to be able to do what I really want to do, not what the grants are telling me to do. So I went into IT, had a pretty successful career in GPU. And when I felt comfortable enough to spend many, many years without the need to work, I dropped everything, went and did my grad work at Johns Hopkins, switched into biotech. I was a professor here and there, and then actually worked for a company that organized conferences here in the United States in biotech. And in a year, we organized more than 30, culminating with a conference on aging in 2008.
Wow, that's early.
Yeah, so at that time I met some of the luminaries who are actually, today there is a Biomarkers of Aging consortium meeting at Harvard, and I met the same luminaries who I met, more than 15 years ago in San Diego at one of the conferences. And I think that the level of progress in this field was not super impressive. Actually, if you look at the timelines, you might think a lot of things have changed, but you get the same statements, repurposing, biomarkers, regenerative medicine. So I was very excited about that field back then. I had very rosy glasses. I always had a feeling that in the next 5 to 10 years, we're going to make a major breakthrough. And aging is going to be a thing of the past, or we're going to get at least on the kind of exponential trend in the field. And by the year 2013, I realized that no. So I actually have to go big myself. And came to the NVIDIA GTC conference, presented, and my presentation was actually focused on, can NVIDIA solve aging? So you can find it on YouTube, and it's actually a fun story because what we presented back then in the kind of nice animation with the explainer and how it's going to all pan out using deep neural networks, and high-performance computing on NVIDIA GPU, it's now a reality, or close to reality, or in some areas we have exceeded our expectations. And I presented, and we went into the finals of the NVIDIA's kind of startup competition, but we didn't win. So another company that was making like virtual characters on video games won, called Artematics. And now the founder of Artematics is actually going into aging research, which is cool, only now. So, but back then I decided that, I want to go all in on aging and AI. And we went into biomarker discovery originally. Biomarker discovery, target discovery, training deep neural networks on fundamental human biology on different data types to predict age, to predict diseases, to derive biological pathways, trying to decipher the aging processes and the processes that converge on this kind of boundary of where aging meets disease, to find common targets and try to sell it to big pharma for almost 2 years until we realized that there is no money in targets. So it's an exciting area. Everybody's talking about it. Everybody wants to do a collaboration in that field, but in reality, there is very little budget for this, and big pharma likes to go after validated targets whenever it wants to make really big bets. So unless you have your own chemistry, you would not be able to realize the full potential of the target. And we decided in 2015, 2016 to bet very heavily on generative chemistry. That was the best decision of my life. So I got familiar with Ian Goodfellow's and Yoshua Bengio's work on generative adversarial networks. In 2014, we started following it. 2015, we decided to go big onto molecular generation with specific features as generation conditions, and started doing POC or proof of concept publications, originally theoretical, so where you do kind of in silico validation, and then started actually validating experimentally. In 2017, we got our kind of first experimental proof that it's possible to generate a molecule in the same way as you would generate, let's say, a flower with additional few petals with high resolution. And then we synthesized and tested. 2018, we published our first papers in that, experimental validation. 2019, we published our breakthrough paper describing the generative tensorial reinforcement learning concept with experimental validation. So showed that we can go from new target that is kind of pulled out of the hat for us with a validation partner. And we can go to mice in 46 days with novel chemistry, just generating 6 molecules. 4 of them worked. One went into mice, 46 days. So we were like, okay, we're onto something. Mm-hmm. And we raised a round. So our first big round, $37 million, that allows us to go into software and also into drugs. So we decided to put the software in the market to ensure that we are always at the forefront. Because if you start working on your own pipeline, if you start working on your own drugs, and you don't have the software in the market, once few of your drugs go even developmental candidate, your software becomes irrelevant, your AI platform. So that I saw many AI companies die like that. So they might continue publishing, but they don't use it internal anymore.
Sorry, why does the platform die? I don't understand.
Because the platform needs to be updated. Every 6 months, you are irrelevant. Because there is new algorithm coming out. Think of you being in ChatGPT world. Currently, well, I guess there is a little bit of industry collusion, but they are not doing the model updates every few months, but they could. And in our field, we also do at least every half a year, most of the models get replaced in my software.
So if it's on a core revenue stream, then it sort of is forgotten, right?
Yeah,
the company doesn't
and the revenue there is tiny. So you have to stay at the forefront, and the revenue is tiny. So if that's your major source of revenue, that's it. How can you compete with me who can sell a drug for $1 billion and then milk it to support your software business? Mm-hmm. So yeah, software business may lose a little bit of money, but then you get massive industry validation. So you've got many companies, many academics using your software, and you protect yourself from their data. So there is a stone wall. You don't want their data is toxic. Mm-hmm. But what you want from them is thumbs up, thumbs down. So if your software, your model worked in their hands within their set of experimental conditions, and they give you a thumbs up, yeah, Alex, we got a developmental candidate. Mm-hmm. Or they say $250,000 spent and we got nothing. You're okay, let us update, get this feedback, we call it feedback. And we use the reinforcement learning from expert feedback that you cannot buy for money. So you have to either do the experiments yourself, which we also do, or send it to a community and have community level validation. Mm-hmm. And believe me, if you open source the software just completely, folks are much less likely to use it than if you make it cheap, but commercially accessible and also with all the bells and whistles that pharma needs in order to deploy it commercially. And it's a very long sales cycle, but once you deliver, you start getting really amazing user base. You enable them. They understand that they can now plug your software into their own pipelines. And if they're really good at AI, they can even connect it to their own models or use your own models for reinforcement learning. However, you always use that same software to develop better and better drugs. And then you can scale, and then you can get to what I like to call industry benchmarks. Because if you're looking at one AI company versus another, how do you compare in drug discovery? Like, what would be your, like, what's your guess? How do you compare them?
How to compare between
AI companies in drug discovery.
The only way is based on the quality of their assets, right?
Yeah, and how do you evaluate the quality of their assets? Like, what are the ways?
Clinical trials?
Yeah, what else?
I'm not sure. You tell me.
So basically, you need to look at several features there. And those are very important questions because actually most analysts, journalists, industry leaders, they usually kind of evaluate the companies by how much money did they raise. That's that blows me away. So even like ChatGPT started doing the same thing. So somebody announces a huge round, and if you say, okay, who is number one? They're like, oh, this one, because it raised more. Like, why? If it raised more, it doesn't mean that it's better. Like, what did it deliver? And then, publications or patents, another like feature. And then people, but all of those are completely irrelevant. What you need to look at is where AI can help you deliver drugs faster, cheaper, with higher probability of success. And that is from zero, from target ID to developmental candidate. So one step before clinical trials. Everything after that is super regulated. So you're basically moving with the speed of traffic. You can, tweak it here and there, say, okay, I'm doing more personalized trials, I'm doing better clinical trial design, I'm doing submissions, review, evaluate the clinical data with AI. But in reality, it's kind of like designing a sports car. So you can test it you can beat everybody from 0 to 100. After that, you're moving with the speed of traffic. So because there is traffic police. IND enabling studies, the reason why they're called IND enabling studies is because there you're doing GLP toxicity studies. So good laboratory practices, toxicity studies, very formal studies that are usually run by a contract research organization that charges you money for it, unless you have a GLP facility yourself, very rare. And there you need to demonstrate, let's say, 28-day GLP toxicity studies in 2 species, like dog and monkey, or like rat and monkey, or rat and dog, whatever the regulator would take in order to give you IND approval to start human clinical trials. So for some drugs, you don't need that, but for most, you do. And that is a regulated process. You cannot fast forward it with anything. The real delta is 0 to developmental candidate. And you need to see how long it takes to go from 0 to developmental candidate, DC, how much it costs you, what is the novelty of the assets that you are trying to go from 0 to DC, because if you're fast following or doing something very similar, just with minor modifications, it could be like significantly faster and higher probability of success. By the way, sometimes commercially viable, so a lot of people do that. And you need to look at how many actually progress to clinical trials. So for us, the typical time now with our current AI and past AI is 9 to 18 months to developmental candidate. Usually for this kind of novelty of the target for industry standards within traditional approaches, it's like 2.5 years to 4.5 years. Sometimes they actually never take novel target to developmental candidate and then to phase 1. Like they might actually, play the same tune for a very long time and then deprioritize it. We'll never know. So for us, it is 9 months to 18 months. 2 examples are published in peer-reviewed journals, so 12 months and 18 months. So both in Nature Biotechnology, high-profile journal. And then we demonstrated that we took 10 to clinical stage, and now there's going to be more this year, hopefully. And we took 1 to phase 2A trial and completed with safety and some promising efficacy within that indication. So we took a drug for IPF, which is a rare disease with huge unmet medical needs. So nothing increases the forced vital capacity over 52 weeks, or like restores it, or even keeps it at bay. And we've demonstrated after 12 weeks that, again, very promising trend. So I cannot make big claims for anything that is more detailed. You can go into Nature Medicine paper that we recently published, where we fully disclose the results of the trial. And there is promising efficacy showing at high dose, 98 milliliter increase in forced vital capacity, the essential measure of lung function that we actually lose during aging. So after like 65, we start losing like 30, 40, 50 milliliters of lung capacity every year. It's just that IPF patients lose it very, very quickly, and without treatment, they lose like 400 milliliters. On current standard of care, they might lose maybe 150 in a year. So when after 12 weeks you show some increase, it's very promising. There have been several attempts to demonstrate increased forced vital capacity, but those never sustained over long term. So we still have a long time to go, and it's very expensive trials, also pretty lengthy. So most people in aging research don't even realize like what it is to be in like phase 2, phase 3 with a novel target, a novel molecule. So it's very lengthy, very expensive, and very few successes. Like even if you look at current stats for approved drugs, it's like 50 drugs approved every year, ballpark, and maybe like 7 innovative. I'm talking about like very innovative globally after spending so much money. So this is a game where most of the time you fail over like 10, 12 year time horizons. So it's not great at all. And what we are demonstrating right now is the preclinical speed records that we're trying to set. And that's where you actually need China. So I actually think that you would not be able to set a drug discovery record like that without China. That is why for us, we are not benchmarking ourselves against other AI drug discovery companies. It's nowadays irrelevant. We are doing that in China, also AI drug discovery companies, because they just call themselves biotech. Everybody is utilizing AI nowadays. And some of the companies there demonstrate in biologics that you can go 6 months to developmental candidate if you are not dramatically different from whatever is out there on the market for that particular target. And sometimes they sell those drugs for a lot of money, for like billions, because they find the right novelty, confidence, commercial tractability kind of optimization, even though it's kind of done using human intelligence. And the work is just so efficient, so high quality that you have to go check it out and compete there. And of course, utilize local infrastructure to be competitive globally. Otherwise, you just won't be able to do it. So we realized that at some point in time in our history, 2019, that we have to have boots on the ground in China and go very heavy there to be able to access the infrastructure, the hardworking, abundant labor, and also having the ability to have gladiatorial battles with the local companies in the kind of China gym, because otherwise you're just, fluffy treatment in the West where you get a lot of money, try to go after a project, and nobody punishes you for failure, saying, "Oh, it's a success, mild success."
Interesting.
Great job. Don't come on Sunday, or don't come on Thursday, Friday.
Welcome to the Frameshifts podcast. where we spotlight the world's most ambitious biotech founders, investors, researchers, and changemakers. This episode is brought to you from the offices of Polsinelli, a national law firm that helps biotech companies do everything from incorporation to patents, trademarks, and venture financings. Thanks to Polsinelli for being part of the growing ecosystem of scientists, investors, and builders shaping the century of biology. So we'll get more into the regulatory side as well. But to relay back to you what I've heard so far, just so I understand, is that the only thing that matters in drug discovery and drug development is this period between sort of target identification and the actual design of the molecules. And the big bet that you made was on generative chemistry.
And generative biology.
Which
is demonetized, yeah.
Talk to me about what did the drug development pipeline look like kind of around the time when Insilico started, and what does it actually now mean for AI to discover the target and also to design the molecule?
Yeah, so our first 5 years after we started were purely algorithmic. So we did not do our drug discovery ourselves. We supported pharma in that, and also kind of did collaboration projects. So we tried everything. So first 5 years we were trying to prove that the tech can actually work, and we're learning. So thank you, our early investors. You didn't give us a lot of money, but we spent it extremely well. And I, of course, spent everything I owned at that time on Insilico. So when we started, we didn't have a pipeline. And the industry benchmarks, I don't think there were actually good industry benchmarks. So we were the first ones, I think, that actually started counting from zero to developmental candidate across like many, because we started doing scale after 2019. It's kind of like OpenAI, right? So if you think about, they started around the same time, promising bright future, and only 2022 they opened up with the real commercial product. But then the Pandora's box was open for everybody, and everybody started racing. So same story for us. At that time, the timelines within Big Pharma for a novel target, novel molecule were like 4.5 years, maybe even more. And at that time, people were regularly synthesizing 2,500 molecules per program in small molecules. So it was like norm. By the way, to put you in perspective, synthesizing one molecule may cost you like $2,500 just to go for a first date with a molecule to see if it binds. And then $5,000 to see if it's active in vitro, and maybe additional like $15,000 to see if it's okay in a mouse for PK. So those are expensive processes, and they actually are pretty lengthy. In our world, once we started drug discovery 2020, we synthesized 79 molecules for our first program that went into phase 2. So that was very few.
Much less.
Yeah.
What is it about generative chemistry that enables fewer candidates?
So it allows you for multi-parametric optimization, multi-parameter optimization. So think of it as, and I'll give you some kind of non-DEI friendly perspective here. So imagine that, so I apologize if I offend anybody, please kind of close the, close this broadcast right now if you're, touchy-feely about those things. But imagine if you wanted to find yourself an optimal wife, and you wanted somebody who is beautiful and knows how to play the piano and behave socially. What would you do? You can do it randomly, Tinder or something like that. By the way, not a bad idea. So do a screen, or go to Miss Universe contest, where they already selected, generated those perfect models, and then you are just looking at, everybody is good, so it's already generated set. with perfect parameters on every level, and then you need to do deeper validation, so to speak, to kind of narrow the set to your perfect molecule. So in reality, if you could just grab just one and marry, again, bad analogy probably, just came up recently. So in that case, you could still win. But if you were to work with this small subset of molecules, because at the end of the day, it's a marriage, right? So once you selected your candidate, you cannot change it, right? So that's why it's a marriage. If you are working with a small subset of already perfect molecules, they are different, they're very diverse, but they will have all the properties you need. And then you start kind of deeper dating, right? So you go for dinner, look at how well it binds. Does it bind better? Does it have any toxicity? Does it have any off-target effects? Does it perform well in different situations? Does it interact well with others? Is it good with your family? So with the rest of the portfolio, can it combine? So you do all those experiments, you select one. After you've done that, you're done.
And I presume also all this experimental data that you generated goes back into reinforcement learning of the models themselves?
Correct.
Yes.
So the new approach that we actually started developing pretty recently, we call it the pan-flute reinforcement learning. So where we take already validated models, sometimes those models may be very small, and they might perform like very simple, stupid task.
This time we're talking about chemistry models.
Yeah.
Yes. Okay.
Okay. Sorry, I'm still on the kind of. I hope we're not still not on the kind of selecting your perfect spouse idea. By the way, some people would disagree with that approach or analogy. But yeah, if you want to come up with a perfect molecule. Sorry, I got steered away. So let me restart for a second. So if you validated one of the models experimentally, and it, for example, produced a perfect molecule that reached a certain level of validation experimentally, you want to hold on to this model and test it several times. If it worked multiple times, maybe you're actually okay just using this model for that particular task. Mm-hmm. And most likely you will continue doing that for maybe the rest of your life because it's just a very good model. Like it knows how to do 3 + 3 = 6 consistently. Or hot dog, not hot dog. As if you know the analogy from Silicon Valley movie series, TV series. So in that paradigm, you validated a very cheap, cost-effective model that performs one task. Now, if I want to teach a very large model that can perform many tasks, that's trained on many, many different diverse data sets, and if I want to reinforce it to achieve state-of-the-art performance within the task that I have the small model working very, very well, I can actually use a small model as a teacher for a much larger model. So you create this gym where you rank your validated models by the level of computational intensity and start teaching your new large multimodal model to perform, to achieve state-of-the-art performance within every task. Mm-hmm. And if it starts failing at some of those tasks, you say, okay, well, I'll just re-engineer this model, retrain, do a different architecture, maybe attach some more kind of physics Lego blocks to it, and try to do it again. In this case, I don't need to spend hundreds of thousands of dollars on compute. I could just limit it to a certain number of tasks where those smaller models can help you reinforce. And then when you lose it, you just redo it again. Sometimes you can go all the way to 3D and get perfectly performing large models, multi-model models that can also do zero shot into tasks that they have never been taught to perform. And that's what I like to call the path to pharmaceutical superintelligence. So we are doing that in chemistry and biology separately right now, more in chemistry than in biology. And now our first model, that is multi-model, is already on AWS being tested for almost a year, called NACH01, Natural and Chemical Languages Model. And now we are working on orders of magnitude more complex models, where we are trying to use this pan-fluid reinforcement learning to achieve, like for me, superintelligence is basically the ability to solve all tasks beyond human expert level at every step of pharmaceutical drug discovery and development, or in all tasks within the chemistry space, better than humans or state-of-the-art models. And it doesn't mean that we would be able to have a magic wand to produce perfect therapeutics, because even if you take the top experts right now, they might not be able to give you 100% perfect drugs. They will still fail, and hopefully they'll fail less. But here we would be able to do it all in one AI model. And as we advance those models, you might be able to acquire new skills. So that's what we are going for. Because in the field of drug discovery, the delta between drug discovery and multimodal in consumer applications or expert applications like text, images, video, voice even, like singing, like Suno AI outperforms me by far, or top experts in music, right? You can basically create an opera within like 5 minutes, and it will be industrial strength with Suno. So how do we achieve that in drug discovery? Only using experimental validation, because in text, voice, imaging, you can very easily validate using human sensory organs within like seconds, microseconds. You can do we used to do Tinder-like systems for molecules, still have some of those, but humans are not great at evaluating molecules. And only experimental validation all the way to human can tell you if your model really worked. And that cycle is very lengthy, and sometimes you cannot compress it. I wish we could. Like if we were to delete some of those necessary kind of regulatory steps that we are taking every time for molecules that may be similar to something that has been already approved, or for targets that are completely, like for GLP-1, I have one case, GLP-1, a small molecule, oral molecule, that is originally based on orforglipron scaffold, but very, very different, like 40% different, but can maybe sustain once weekly dosing. So that's the feature that we're adding. So very low clearance and improved safety. That's another feature that we're trying to, even though Orpho is very safe already. So, and imagine if I want to put something like that into humans to measure PK to see if it can sustain once a week dosing, I actually need to go through the entire 9 yards and do the non-GLP tox, 28-day. I did 4 species. And then you need to do GLP tox in 2 species at least. Sometimes you do more just in case. And then go human, submit the IND, go human. So between the non-GLP tox and human, we're talking about like 14, 15 months.
And we should also clarify here, we're talking about GLP-1s, but also good laboratory practices as well.
Yeah, so GLP tox,
there.
That's good laboratory practices terminology. Thank you for expanding it. But GLP-1s we've all seen on South Park, so we probably know what it is. So Ozempic-like molecules, but now small molecules. It's a new trend. So imagine that Ozempic in a pill once a week. That's easy to store, easy to ship, and also accessible to any population group, regardless of their wealth. So cheaper, and you dose it only once a week, maybe combining with other drugs. You do need that. And it's also a very commercially viable area. So if you have a really good drug in that area, pharma companies really want to buy it. We already have many interested parties in that particular drug, even though we're very early. It might be more interesting for them than a novel target in phase 2. Believe it or not, but that's the reality of pharma. They like validated targets with differentiated chemistry.
Okay. So I think this gets at something that the industry and biotech in general has been quite obsessed with over the past maybe a couple of years since the ChatGPT moment, was how do we get biological multimodal models to become as good as sort of these large language models are getting at creating text. And the problem, as you mentioned, is that humans are actually quite bad at reinforcement learning or reinforcement training of these models, because most of our historical biotech drug development has been sort of trial and error. We throw millions of molecules or hundreds of thousands of molecules in these high throughput screens, and they're very expensive. So we're not actually very good at guessing what the chemistry should be.
Yeah.
Right. So hopefully now, with the advent of these great algorithms that can train on data, we can use experimental validation to feed back and reinforcement learn of the models. But the problem has always been sort of closing the loop all the way, right? So you're big on lab automation. You obviously, Insilico also was doing like Insilico hypothesis generation, Insilico design of the actual chemistry of these molecules. But like, what's the missing infrastructure that can really make this reinforcement learning a full closed loop?
Experimental validation, and you cannot go around that. So that's why I think that this pan-flute reinforcement learning approach that we're developing now, where we validate smaller models that can really do the job experimentally, and then use them to reinforce larger models, that's why it's so valuable. Otherwise, again, the disconnect between the time it takes you to develop a model, very capable model, and the time you'd validate it to at least the lowest possible denominator for validation, developmental candidate. Of course, ideally you want to show phase 2 clinical trial complete. So if you don't have that, actually pharma companies won't even believe you that you have the capability. But the delta between 0 and phase 2 complete, and I've demonstrated, is close to 5 years. And I've done it very quickly, so with a novel target, and published at every step of the way. So everyone in the large language model world who runs a large kind of quote-unquote superintelligence company, nowadays many of them came out and said, look, we're going to cure all diseases in 10 years. Okay, I used to say that too in the early days of deep learning. We're going to double our lifespan within the next 5 years. I've heard that too on stage from like a major player in the field. And you realize, wow, that's a nice statement to say. But if you actually try and go and apply that to biology right now, you're going to fail most of the time, even in target discovery, even in experimental validation using lab automation. So there you're going to still fail most of the time if you're going to go after novel stuff. Experimental validation is a limiting factor for everything. And right now, we just don't know enough about fundamental human biology to be able to intervene at levels where we could cure all diseases. Not with gene therapy. Ideally, that would be the proper approach. Not with even antibodies. And now we've got perfect tools for antibody design. Those are usually really, really nice Lego blocks for a big puzzle. But so far, nobody created the big kind of Lego puzzle all-in-one model that could develop super intelligence to produce one drug that the person could take and get younger or cure cancer. Also, to be able to test something like that, right now there is no country in the world that would allow for something like this. Even if I want to take this, my supposedly safe GLP-1 small molecule that is following a safe scaffold with additional safety overhangs, if I want to synthesize it and test it on myself, it's kind of illegal, big taboo. Trust me, I asked many times. If for those of the people who know me, I am very in favor of trying things out quickly. But compliance is compliance. I cannot go over compliance. And experimental validation is the massive gating factor. So I think that so I'll tell you a fun story. So we are trying to develop a biological superintelligence model for a very long time. So we actually started with aging clocks, biomarkers of aging, trained on many, many different data types. So there are very few data types where I have not published a predictor of biological age. I even published few on psychological aging features. So basically surveys from massive numbers of people taken over the course of their lives. So I can basically ask you a set of 20 questions and I'll guess your age. And those questions will not be related to your education or kids or anything like that, just psychological. You can actually do that. I can guess your age from a facial photograph. I can do it from a retinal scan, from a blood test, from microbiome, from a stool sample, within a certain margin of error. So there is an entire school now on aging clocks, how to apply them clinically, how to use them in clinical trials, how to use them for mortality prediction, disease prediction, many, many other things. So we decided, well, why don't we build a multimodal system that can take any data and predict your age and maybe also do a few other things. Started publishing a few years ago. So our first one was Precious 1 GPT that could do age prediction and target identification in aging and disease. Then we progressed to Precious 3 GPT that could perform multiple tasks. And there we actually started pausing because validating those performance and tasks is dramatically difficult. And we also realized that once you introduce the the larger numbers of tasks, the model starts collapsing. And you start performing below state-of-the-art performance within the tasks that the model knew how to perform before. So now before doing our Precious 4 GPT, which we kind of have internally, but we haven't released it and we're still working on that, we're actually thinking about how to constrain the number of tasks and how to ensure that we don't lose performance within those tasks. So we're making models even more specialized again. Before we would increase the number of tasks. We're increasing the complexity of those tasks. And now I'm actually exploring a new approach and a new business model. So if anybody is listening here from large language model companies that are specializing in consumer-driven LLMs, like ChatGPT and Anthropic, Claude and X, Grok, or Gemini, even though technically it might be a competitor nowadays with their isomorphic. But if they're interested, I would still be collaborating. So now we can actually utilize my experimentally validated models, the zoo of models that have achieved state-of-the-art performance and have been experimentally validated. Now I'm actually thinking about approaching some of those LLM vendors to train their large models on chemistry and biology, utilizing the intelligence acquired by my smaller models, so that everyone in the world could acquire the same capabilities in biology and chemistry. So demonetize it completely. At the end, I think somebody will do that. Maybe us, if we don't get a collaborator, we'll have to do it ourselves, or partner with one of the DeepSeeks of the world. There are now multiple, and I think at the end it's going to be the infrastructure that will decide the winner. Capital and infrastructure. Intelligence will be equal for most. I'm talking about target discovery and small molecule chemistry. So there we would have more shots on goal. And their efficiency of execution will be the key parameter in biology and chemistry. I think it's like maybe 4 or 5 years from now. But you would still be constrained within the same regulatory frameworks.
So when you're talking about infrastructure, obviously part of that is fully automated, robotic experimentation. How are you thinking about that? I know you have a factory, or like a facility in Shanghai and one in Suzhou. Talk to us about that.
So I think that automation, specifically in biology, is extremely important. And chemistry automation will be commoditized because there are so many contract research organizations that are investing in that. So we decided not to even compete there in chemistry automation, because chemistry automation is like you need to have really high throughput of chemistry in order to compete in this field. And only contract research organizations can actually do that. But in biology, you could actually win as a single player in the field, like as a single user of your own technology, and then you can choose to open it up for others or not. And we developed a fully automated facility that can help you do target discovery. So throw in a sample, and the system will automatically identify targets, select the validation workflow, and validate using imaging, multi-omics data, and many, many other approaches. So CRISPR, RNAi, whatever model.
And you have like humanoid robots as well, just walking around.
We're experimenting with them.
That's pretty cool. Can you tell us a little bit about sort of how effective this experiment has been, and like what actually are their capabilities?
Currently not super successful. So they can give you a tour of the lab. We're not developing our own humanoid platform. Okay. So the beauty of deep integration into the China ecosystem is that there people are extremely collaborative, especially in the early days, because everybody is competing with everybody. It's like a gym, like a massive gym. So there is like 100 humanoid companies there. So we chose one of the best ones, took their platform and started focusing on intelligence. So how to really develop the embodied AI for the lab. And for that, we're generating the data set to be able to actually train properly and execute on more sophisticated laboratory tasks than just walk around, pick something up. Those tasks are already done, but those can be performed by expert human. And what we also realized is that in many cases you don't need a humanoid, or you need like just a platform that drives around, because actually in a lab you don't even need bipedal, right? Because there you need micrometer precision. You need to have very low vibration. So any kind of vibration will be detrimental to your cells, to your chemicals, to your reagents. So it's a good idea to have something that is very smoothly driving, but with human-like infrastructure that allows you to manipulate, to perform basic laboratory manipulations.
Basically hands.
Well, sometimes with a head, because nowadays you can buy platforms that are cost down. So it might be comparable in cost to a humanoid, but they will be even more stable and more fit for industrial applications. I think at the end of the day, maybe like in 5 years, we might be able to get the vibration frequency to the level where vibrations to the level where it's compatible with biological work. In the ideal world, you want to have minimal vibration. You want vibration only when you're shaking something. It's great for a specific purpose, but you also want to make sure that everything is smooth flowing and again appropriate for lab. And currently we have autonomous guided vehicles already that are driving around. So just making them more capable would already make the lab much more capable and ensure that humans don't go into the areas that require that have very high levels of sterility. So you actually need to ensure that there are no contamination both ways in the microbiology lab, for example. And you want to make sure that most of the kind of human tasks are performed very consistently. And with a humanoid platform, you can do pipetting consistently. I'm not sure, like you definitely have done pipetting and you know how to do PCR, but when you are doing the aliquots, sometimes I forget if I pipetted into the well or not. It's just such a tedious...
The eternal issue of humans,
Okay, how many
every
milliliters?
week.
And you have to do many of them, right? So if you are doing some of that using humanoid platform, it's beautiful. And also, you don't need to then over-automate the lab and utilize the equipment that has never been designed for lab automation, right? Because some of the equipment vendors, they never, like even those that focus on automation, they still want you to have like an LED display, LCD, maybe touch screen with buttons. Everything has to be manually loaded. And then they assume that, since you have to do this step once every 48 hours, they're okay with, manual loading. And if you say, guys, like, how about I put additional automation equipment for, opening this door and loading this reagent? Like, no, we won't even be able to sustain your warranty if you do this. External arm, right? So I just drill it a little bit, right? And here I put like an additional compartment. No, you cannot touch our equipment or automate like this. But if you have a platform that is fully compatible with human, you can do whatever you like.
I see. Okay. Yeah, I mean, clearly there is this receding space where humans are going to continue to be valuable in biology, right? So it's the pharmaceutical superintelligence concept is first of all fascinating. So just to relay that back for the audience, you have these smaller experimentally validated models that are each good at like a particular task. Like for example, target identification or the design in maybe a particular sort of chemical family. You can expand on that a little bit as well. And then you use this to train sort of biological language models on these validated smaller models, and this way you have sort of pharmaceutical superintelligence and also biological superintelligence. There's a lot of companies that are sort of touting this concept, right? So there's Lila, there's sort of Basecamp, there's obviously your company, which is kind of predates most of them. What does the end state look like? Like in 5-10 years, when I log on to insilico.com/biological superintelligence or pharmaceutical superintelligence, what is the interface? What can it do for me? What do I type in? Like how intuitive does this become?
Well, in the ideal world, you shouldn't be doing that with Insilico. You should be able to use your favorite ChatGPT for that. That would be seamlessly connected to my infrastructure. And you would just tell your favorite assistant to design a drug with the desired characteristics. Or in the ideal world, you would just take a drug off the shelf where I already have a molecule. Because most likely by that time I'll have many starting points and programs. And I would just be able to say, okay, I've got enough cash, here you go, continue. And that's what the pharmaceutical companies should be doing. And I think they will be doing that. So not exactly significantly cutting their R&D, but making it so much smarter where you actually start with a validated starting point. So validated target or pre-validated target, pre-validated molecule. So the way I see it, you can either talk to me or to your favorite LLM and say, I have a market need where I've got this specific type of head and neck cancer. This particular population group, get me a small molecule drug, oral, with this toxicity profile. I'm willing to pay this amount of money. And I will look through my databases, also maybe partners' databases, tell you, okay, you can actually buy it right now for this amount of money. And if I don't have one, I'll say I can make you one for this amount of money. So it becomes, again, a game of infrastructure, so the ability to execute, and capital. So I think those two are extremely important. That's why we're going now into, for example, Middle East, and I'm attempting to design a novel drug within our current timelines in Abu Dhabi, for example, with just 4 people. So 4 people responsible for the entire drug discovery program of moderate novelty, and I'm trying to beat the 18-month timeline to developmental candidate and then license it out. Because remember when I asked, okay, how do you measure success? So yes, you can actually complete a clinical trial and get the drug approved. That would be the best thing to do. But the second best is actually to license your drug to an expert company that is willing to pay a lot of money for a high-quality therapeutic. So imagine if you do that in Abu Dhabi and so then license that to an external party, then everyone in the region will realize, actually, that's a money-making business. And it's basically like invest, get the money back quickly, or if you want to go next step, let's do local clinical trials with our own locally discovered drugs. So the idea is to bring more people to the industry in areas where people even never tried to aspire to discover a novel therapeutic. Like imagine doing that in Africa. Like if one of the African countries wants to become a new Singapore, and of course, as you know, Singapore was a dictatorial regime where things were kind of properly structured from the very beginning, and some of the decisions were made that would not appeal to a democratic community. And somebody says, you know what, we are going to focus on drug discovery, and we are going to become great at it. And we're going to work with Insilico or some other company to deliver not only developmental candidates, we want to deliver like ready drugs that are clinically validated in our country. Just redo the trials somewhere else and it's done. We're willing to accelerate. Beautiful. Let's go, guys. You can fundraise for that. You can have seamless AI. All you need is infrastructure and capital. And even infrastructure, you can rent it for capital. So you can convert it. And then locally, you will provide some infrastructure, for example, clinical trial infrastructure. You want to open up your population to clinical trials? Let's go. Australia demonstrated how to do that. Now in Australia, you don't need to file for IND to do the phase 1. And now, like many young people who are participating in those clinical trials for safety, they are extremely familiar with drugs because they are basically selling their time for volunteering for that, but they get to learn about drugs. So imagine when those young kids, usually it's college folks, who get their PhDs, MDs, and go into drug discovery, I would like to hire all of them because they know the safety profile of so many drugs. And they felt it themselves, right? So those are beautiful.
It's so easy to enroll in a clinical trial in Australia. It seems much faster, just intuitively.
Anybody from Australia listening, kudos to your government. That's the way to do it.
Yeah, absolutely. We're running out of time. I've got like maybe one more question for you, which is kind of more of a meta question. We're automating away a lot of biology. And you mentioned that you want more people to enter biotech. You want more people to care about drug discovery and sort of aging in particular. But if we're also automating away all of the things that humans could be either intellectually or physically useful for, what is the case to be made? I mean, like how does one do science in an age of robots?
Again, I think that biology, chemistry, you can automate all you want. Experimental validation and also just the attempts. So somebody needs to run it. You need to, there is a lot of infrastructure, a lot of labor, a lot of human component that is going into this. What I noticed, and I made a few very stupid statements when I started the company. I was actually making statements that we're going to replace some medicinal chemists. Mm-hmm. Oof, super stupid question, statement. In question, can we replace them? That was actually one of the papers that I published a while ago. Can AI replace medicinal chemists? Those are some of the smartest human beings in the world. So if you want your kid to go into some really cool specialty, medicinal chemistry is the one. They'll be able to, it's like an MMA fighter in drug discovery. We hire probably more medicinal chemists than a traditional biotech right now because they help us validate AI, they help us develop more AI, and they help run programs. And they also tell you whether the quality of the product is good or not. I don't think that within the next 10 years we'll be able to go around massive experimentation. We probably will have to do more of it to get to learn biology from first principles. Currently, you cannot reconstruct biology from first principles, even if you simulate the world on a quantum level right now. There's no compute to be able to simulate a human body or even like an organ.
Or a cell. I think we're still orders of magnitude of computation from at a planetary scale from being able to simulate a single cell.
Yeah, well, let's hope so. I mean, we are working on a virtual cell as well. Everybody is. We might have achieved that goal already in some areas where we are doing target discovery. And in some disease models, in aging, for example, we understand replicative and chemically induced senescence really well, at least at Insilico. And relevance of multiple of those processes to diseases. But I think we're very far from solving the problem. And my challenge is that we might get into this perfect utopia where people's attention is focused on something fancy like, TikToks or something else that's very addictive, or Mark Zuckerberg comes out with like perfect AR glasses that put you into the fancy world without trouble, or Elon disables your pain receptors through Neuralink. And we get into this utopia where people don't care about aging and diseases, don't care even more than they care today. And that is the problem. So even right now, like how many people in biotech, you probably know, like most of your friends are probably in biotech. And all of mine, except for those who are in AI. But if you take an average person on the street, or those people who are now, protesting here in Boston, if you look at what they're protesting against or for, it's not about like curing diseases or aging, right? It's about some minority somewhere here or there, somewhere. Maybe it is important, and it's important to do this, but it's not more important than fighting against death. Okay. 100% death.
Alex, it's been a pleasure having you on. I've got one final question for you. What is a book that you'd recommend to our audience that you think will inspire them or teach them something?
I cannot recommend just a single book. I have to give you like a book list.
Give me two. How about that?
Okay, well, if for the audience that is not very familiar with aging, read David Sinclair's Lifespan. Very important. I would say that the kind of some scientific component of this book could have been expanded, and you probably will need to go to a few conferences to learn more. But this is the best book in aging I've ever read in terms of like, why do you need to do this? And some very basics that you need to learn. And other books, I actually recommend learning about the past of drug discovery and reading A $1 Billion Molecule about the history of Vertex Pharmaceuticals, here the Boston multi-unicorn, and the early days of the company. Because what was relevant in the book back then is extremely relevant today. And the delta is 25 years.
Okay, fantastic. Those are great recommendations. I've read both. Obviously, appreciate your time, and I think it's been a fascinating discussion.
It was a pleasure being here. Yeah, thanks for this opportunity.